All right, scholars. So here's one of our labs of the unit 2, cells and biomolecules. We're going to be talking about nutrients and indicators. All right. So our aim says, how can indicators tell us the type of nutrient present? Make sure that you are taking notes because you will have a Google form to fill out. That's pretty extensive. That's going to count as a lab grade. All right. And that lab grade is 20% of your grade. So don't miss anything. All right. Aim, how can indicators tell us the type of nutrient present? All right. So we see the word indicator here. What do we think the word indicator actually means? Well, one of the words that we see in indicator is indicate. When something is indicated for, you're implying that it's there based off of something else. So an indicator tests for the presence of a substance. So an indicator tests for the presence of a substance. In today's lesson, that substance is going to be a specific nutrient. Now, our 4 nutrients that we're going to be using is glucose, starch. So glucose is a simple sugar, starch is a complex sugar. Another nutrient we're going to be using is protein. And the last nutrient is oil, which is going to be our lipid. Okay, so that's pretty much the lab that we've already annotated. Let's go through. So what is a chemical reaction? I'm sure you guys have seen chemical reactions occur, or maybe even done some yourself. But a chemical reaction essentially is a reaction between two substances. But it's irreversible, which means that you can't undo it. So it's an irreversible reaction between 2 or more substances. Okay, so here's some examples of chemical reactions that I'm sure you guys have seen before. The apple core turning brown when it reacts with oxygen in the air. You can't undo that, right? Frying an egg, can't unfry an egg and get it back inside the shell. Burning wood, right? Or burning like matchsticks or something, burning paper. You can't get the wood back after you've burned it. That's a whole chemical reaction, combustion to be exact. And then fireworks, of course. You can't undo a firework and get it back inside of its original state. So what other examples of chemical changes can you think of? So let's read an introduction. Keep that in mind. Nutrients may be classified as carbohydrates, all right? So nutrients may be classified as carbohydrates, sugars and starches, lipids, fats and oils, and proteins. Carbohydrates are a group of organic compounds that contain the atoms of carbon, hydrogen, and oxygen in the general formula CH2O, N, subscript N. The biological process of respiration, that's going to come up again, breaks down the carbohydrates to yield energy for plant and animal activities. Lipids provide organisms with a concentrated source of energy, yielding twice as many calories per gram as carbohydrates or proteins. Proteins are complex organic compounds formed from long chains of amino acids chemically linked together. In this exercise, sugar, starch, fat, and oil and protein will be tested using indicators, all right? So these are our nutrients, and then we have our indicators, all right? So what are indicators? In food analysis, indicators are used. Indicators are used to detect the presence of specific organic or inorganic compounds, such as sugars, protein, starch, glucose, or oil. All right? So I'm gonna start my video. All right, I hope you guys can see me. All right, so here we have glucose, which if you remember is a simple sugar, it's a monosaccharide. All right, that's one of our nutrients, glucose. Our second nutrient is starch, which is a complex sugar. And if you can see, the starch is actually very much settled at the bottom. So you need to shake, shake, shake. Shake, shake, shake. Shake your starch up. All right. That's starch. Then you have protein. And yes, it smells as bad as it looks. All right. And then you have oil. All right. If you remember, oil is a type of lipid, doesn't mix well with water. All right. Now for glucose, we use a specific indicator, which is known as Benedict's solution. Okay. So Benedict's solution tests for glucose. We have indicator, whoops. And nutrient. So our nutrient for glucose is glucose, and our indicator for glucose is Benedict's solution. Pretty good, huh? All right. So those are those two, glucose, Benedict's solution. I'll put that off to the side. Next, we have our starch, which we just shook up. And what we use to test for the presence of starch is iodine, AKA Lugol's solution. So iodine / Lugol's solution. All right, so I'm gonna write that there. The Lugol solution. Test for the presence of starch. Next, we have our protein, which is here. And we actually have to mix two substances together in order to get what we need. We have to mix KOH, which is actually a base, potassium hydroxide. You don't need to know that. And we also need to mix CuSO4, which is copper sulfide, together, copper sulfate, excuse me, together, and together, those are going to test for the presence of protein, all right? But what we call that indicator is Biuret, B-I-U-R-E-T, Biuret solution, and it tests for the presence of protein. And then last but not least, this one is probably most familiar to you guys, oil. And we use a brown paper bag, right? So last but not least, we have oil, or sorry, brown paper. It's gonna test for the presence of oil, which is our lipid. Okay? So let's actually begin and see what this actually looks like, okay? So we're gonna start with glucose and Benedict's solution. Now, I have a test tube here, and I have my Benedict solution. And I'm just gonna put enough drops for me to actually see it, right? Not really expecting anything to happen because I'm simply just putting Benedict solution in here, all right? So we see Benedict solution, which is here, and then we have our glucose. Now we're gonna see what happens when we add glucose in here. Ready? So it started out blue, and it's still blue. But we thought that Benedict solution tested for the presence of glucose, so surely we should have seen a color change or something happen. We didn't see that happen because this is a special test. Benedict solution, a test for glucose, must be heated up on a hot plate. So I know you can't see it, but I actually have a hot plate on with a beaker filled with water that's boiling or about to boil. And we're just gonna let that sit in there while we conduct our other tests, okay? So we're gonna wait for that test, wait on the results of that test and see if it actually tests for the presence of that. Next, we have our iodine. We're just gonna put just enough iodine to see inside of another test tube, inside of a clean test tube. Okay. And as you can see, it's like this really pretty brown color. That color that we call that is amber. Okay, A-M-B-E-R, amber, all right? Now we're gonna see what happens. We're gonna shake it up again. It's starting to settle. We're gonna put a few squirts of starch in with the amber colored iodine solution that we just put in here. And we're gonna squirt it in. If you can see that, you'll literally see it turned black, right? So it turned black. So if you look on your paper, you go where it says iodine solution, it'll literally go from an amber color to black if starch is present. Let me show you guys one more time what it looked like, okay? Now I know it kind of doesn't look black wholly, but it is. I'm so sorry, guys. There's literally a squirrel on the thing, and I really don't have time for this squirrel trying to get inside of this classroom right now. All right, our glucose is looking pretty good. Let's move on to the other test. Now, next is protein. And remember, we had to mix two different solutions for two different compounds. So we're just gonna put a few drops of copper sulfate. That was a little more than a few, huh? And then a few drops of potassium hydroxide. Okay, mix, mix, mix. And it actually makes this really pretty blue color. You guys see that? So where it says Biuret solution, it'll turn from blue. We know that. Let's see what happens once we actually add this stinky protein. Ooh. Whoa. Ew, 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 ew. All right, let's add some protein. It still looks blue. So what I'm actually gonna do is add a little bit more KOH. Ah, there we go, there we go, there we go, there we go, there we go. This KOH is pretty weak in concentration, but you guys should actually see it turned like a really pretty purple color, all right? So we call that violet. So under Biuret solution, it goes from blue to violet, which is essentially just supposed to be a light purple if protein is present. Okay. All right. Now, last but not least, we have our oil, or last but not least until we go back to our glucose test, right? We have our oil, and we have our brown paper bag. No need to get messy with the oil. You'll be able to see a change almost immediately with just like a dab, right? Right? So for the brown paper test, a stain will remain on brown paper if fat or oils or lipids are present in the food, right? So that literally goes from, these are the words that you'll use. It goes from opaque to translucent. Not to be confused with transparent. Transparent means completely see-through. Translucent means kind of see-through. So semi see-through. All right, let's check on the Benedict's test, which is what we're here for. So remember, if you remember, it started out blue when we put the Benedict solution in there. Then we put the glucose in there, nothing changed. All right, I'm actually gonna add a little bit more glucose. I actually added a lot more glucose. We're just gonna wait and wait and wait a little bit more. So for Biuret solution, if protein is present, it goes from blue to violet. For starch, if starch is present using iodine solution, it'll go from an amber color to black. For the brown paper test, it'll leave a stain and it'll essentially go from opaque to translucent, which means semi see-through. Okay. And here is our glucose test. So I left it in there a little bit too long. I did. I'm not gonna lie to you. But essentially, it turned orange. But Benedict's test is really special because it goes from blue to either red, which indicates a ton of glucose is present, to orange, which means that there's a good amount of glucose present, to yellow, which means there's some glucose present, or green, which means that very little glucose is present. So red, orange, yellow, or green, okay? That might come up again. Here, all right? You're gonna complete the questions there, and will count as a lab grade. So make sure that you get that done. All right. All righty. So thank you guys for everything. I hope you learned something. And yeah, study, study, study.